My name is Maranga from the Malposca Center of Biotechnology. I'll be presenting a poster on the comprehensive functional annotation of metagenomes using deep learning-based method, deep function residue identification. This project is a collaboration between Malposca Center of Biotechnology, Poland, the Flatiron Institute in the US, and the Liggins Institute, University of Auckland, New Zealand. I will start by giving a brief introduction of the human gut microbiome. The human gut microbiome is associated with various diseases, including type 1 diabetes, ulcerative colitis, and many others. However, the exact mechanism of how the gut microbiota interacts with the host-caused disease remain poorly understood. Mainly, this is due to our limited understanding of the function potential encoded in the complex gut microbiome, as currently only 50% of the microbial protein coding genes may be functionally annotated. In our current study, we aim to characterize the functional potential of human gut microbiome in type 1 diabetes. We used the DIABIMMUNE infant gut microbiome cohort. This data was previously collected in Finland, Estonia, and Russia. We have developed a custom functional annotation pipeline based on DeepFRI, which is a deep learning method that uses graph convolution network to functionally annotate proteins and outputs the gene ontology probabilities. Our method integrates de novo genome reconstruction, taxonomic profiling, and functional annotation. I will briefly take you through our pipeline. We had shotgun metagenomics data coming from the DIABIMMUNE data of around 1,067 samples. We performed de novo genome assembly using megahit to generate contigs. We further binned these contigs into metagenome assembled species using MetaBat and taxonomically profiled metagenome assembled species using genome taxonomy database. Further, we predicted genes using Prodigo and clustered these genes based on sequence identity at 97% using CD-HIT to generate our non-redundant gene catalog. For the functional annotation, we used DeepFRI, CNN-based DeepFRI. We also benchmarked DeepFRI using EGNOG, which is an orthology-based method for protein function annotation. We have generated some preliminary results for analysis. For the assembly, we assembled around 17 million contigs from the DIABIMMUNE data. And our non-redundant gene catalog consists of 1.9 million microbial genes. For the annotated genes, with DeepFRI molecular function at a threshold of 0.2, we're able to annotate around 1.8 million genes from, that is 99% of our gene catalog. We compared this with EGNOG gene ontology, but we were able to annotate around 15% of our gene catalog, that is 280,000 genes. We also considered other function information coming from EGNOG free text description. This is annotation information obtained from the source database. With EGNOG free text description, we were able to obtain information of around 1.3 million genes, that is 72% of our gene catalog. And now looking into different gene sets annotated by DeepFRI and EGNOG free text, our results show that roughly there is 1.3 million genes that are shared between DeepFRI and EGNOG free text. And there is around 528,000 genes that are unique to DeepFRI. And by comparing gene sets annotated by DeepFRI and EGNOG gene ontology, there was roughly 279,000 genes that are common between DeepFRI and EGNOG gene ontology. And there is 1.6 million genes that are unique to DeepFRI. We see that by considering other information coming from EGNOG free text description, we are able to increase the number of annotated genes. And since a protein may have multiple gene ontology, making it difficult to accurately describe its function, we further filtered our gene ontology terms only to include 614 informative gene ontology terms. And now looking into the annotation coverage, this plot shows proportion of gene abundance with functional annotation weighted by relative abundances. On the y-axis, it's the relative abundances in copies per million. And from our results, we see that DeepFRI is able to increase the annotation coverage in comparison to EGNOG, which had an annotation coverage of less than 20%. And including other functional information coming from EGNOG free text description, we see we are able to increase the annotation coverage at metagenome level to up to 80%. To further understand the gene ontology terms being predicted by both methods, we further performed gene ontology level differences between DeepFRI and Eggnog. This plot shows topmost gene ontology terms predicted by both methods. And the green bar shows gene ontology terms exclusive to DeepFRI, and the orange bar shows gene ontology terms exclusive to eggnog. The blue bar shows gene ontology predicted by both methods. We see that in some cases, DeepFRI is producing more gene ontology terms in comparison to eggnog. And one of our open questions is to determine the biological significance of these gene ontology terms. To further obtain an understanding of the functional capacity within the core and the accessory genome, we performed pangenome analysis. Here, we only included 42 species that contained more than 10 genomes. Our pangenome consisted of 73,000 core genes and 358 accessory genes. We further annotated our pangenome using DeepFRI, and our results shows diversity in functional enrichment between the core and accessory genome, where we were able to annotate 25% of the core genes and 16% of the accessory genes. So far, we presented a method for metagenome function annotation with higher coverage. And our next step would be to expand this annotation to incorporate 3D structured DeepFRI predictions. I would like to thank our collaborators who have been involved in this work, especially our collaborators from the Broad Institute, the Flatiron Institutes, Liggins Institutes, and the Malopolska Center of Biotechnology. Thank you all for listening.